Meselson and Stahl's experiment. Matthew Meselson and Franklin Stahl were the scientists who were doing research on models of DNA replication. They were awarded Nobel Prize as well as they concluded that DNA replicates according to semi-conservative model. So what is this semi-conservative model according to which DNA replicates? In semi-conservative model of DNA replication, for example, this is a parental DNA molecule. When it undergoes replication, parental DNA strands separate and will act as template for the synthesis of new strand. So here a new strand synthesized. So in semi-conservative model, both daughter molecules have one parental strand and one newly synthesized strand. This is known as semi-conservative model of DNA replication. Meselson and Stahl used two isotopes of nitrogen N15 and N14 in the medium of bacteria, and these two isotopes of nitrogen can be distinguished according to their densities by centrifugation in cesium chloride solution. As N15 is heavy one, as it have high atomic mass, and N14 is the lighter one, it is less denser, so we can distinguish according to their density in centrifugation. Meselson and Stahl first grew bacteria in N15 medium, and then they did centrifugation. They spun the bacterial sample at very high speed in ultracentrifuge for many hours in cesium chloride solution. And after centrifugation, what they found bacterial DNA strands heavier because they incorporated N15 isotope of nitrogen, heavy isotope of nitrogen, into their nucleotides during their replication. So you can see that bacterial DNA strands both consist of heavy isotope of nitrogen N15 in it. Next, bacteria grew in N14 medium, and then scientists did centrifugation and they found bacterial DNA strands at the top of the tube because these bacterial DNA strands appeared lighter. They appeared less denser as they incorporated N14 isotope of nitrogen, which is lighter one, during their replication. So here you can see that both DNA strands have N14 isotope of nitrogen, lighter one, in their nucleotide. They shifted the bacteria from N15 medium to N14 medium. Now bacteria were shifted from N15 to N14 medium and they allowed to replicate. Scientists took the sample at 20 minutes and did centrifugation, and in centrifugation they found a hybrid duplex at the middle of the tube. So what they found, that this is a hybrid duplex. One of the strand have N15 isotope of nitrogen in it and one of the strand have N14 isotope of nitrogen in it. So this bacterial DNA molecule which were shifted from N15 to N14 medium, they have both N15 and N14 in them. So this is a hybrid duplex. So how they replicates? Scientists came to the conclusion that bacterial DNA replicated according to semi-conservative model. This is the DNA molecule in N15 medium. So when it shifted to N14 medium, they replicated. So how they replicate? First, these strands separated out like this, and then these strands act as a template for the synthesis of newly synthesized strand, new strand. So here, new strand appeared and form a duplex. So you can see this is a hybrid molecule. This is a hybrid molecule. It have both daughter molecules have one strand having N15 isotope in it and the other strand having N14 in it. So this showed that this bacterial DNA replicated according to semi-conservative model. Then as they shifted the bacteria from N15 medium into second plate as well, which also contain N14 medium, and here they took the sample at 40 minutes. So here the bacteria completed almost 2 rounds of replication, and now they did centrifugation, and after centrifugation what they found one hybrid duplex and one light duplex. So let's again these two daughter DNA molecules, when they undergo second round of replication, first these strands separated out like this. So these 4 strands separated out, and then these strands will act as template for the synthesis of new strand. So here new strand will attach to each parental strand. Like this. So you can see that after second round of replication, these two DNA molecules have N14 isotope of nitrogen in it. They appeared lighter, so they appeared at the top of the tube here. And those DNA strands, those DNA molecules which appeared as hybrid duplex, they appeared at the intermediate level because they have DNA strand of N15, one DNA strand composed of N15 and other DNA strand composed of N14. So here one intermediate and one light DNA forms. So that's all about Meselson and Stahl's experiment. If you like this video, then do like and subscribe this channel.